This lesson is about oxidative phosphorylation, the fourth stage of cellular respiration. This stage produces the most ATP and it takes place in the inner membrane of the mitochondria. Now, to understand oxidative phosphorylation, you have to understand the anatomy of the mitochondria. So, here we have the inner membrane highlighted in white, okay, and the matrix, which is the interior, in yellow, and the outer is the outer membrane, as you can see here. This is the outer membrane. And then here, all this here in pink, in between the inner and outer, is actually the intermembrane space. And the mitochondria takes place in the inner mitochondria, but both the matrix and the intermembrane space are involved. So, there are two parts of the oxidative phosphorylation stage. The first part here is the electron transport chain, which is this part, and the second part is ATP synthase. And now, as you saw here, it's very folded. The mitochondria inner membrane is highly folded, and that's to fit many ATP synthase and electron transport chains in it. And imagine if it was just round like this, it wouldn't fit as much, but this folding allows it to fit more. So, about the electron transport chain, it's a collection of membrane embedded enzymes. Well, these are embedded, complex 1, 3, and 4, but complex 2 is bound to the membrane. And there's also electron carriers. So it has these protein enzymes and 2 electron carriers. Okay, so complex 1, 3, and 4, which are involved in taking the electrons from NADH and passing them through. And also you have complex 2, also called succinate dehydrogenase. This should sound familiar. Remember the Krebs cycle in step 6? It was the enzyme, also the enzyme used in step 6 to turn FAD into FADH2. So this enzyme is also used in the Krebs cycle. Okay, take us to the larger picture. So, first of all these energy carriers that were made in the Krebs cycle are sent to the electron transport chain, and also the energy carriers from glycolysis and pyruvate oxidation as well. So the most were made in the Krebs cycle, 6-NADH2 and 2-FADH2. And once they're sent there, what they do is they give up their electrons. Electrons are energies, so complex 1 takes the electrons from NADH, converting it back to NAD+. Here, it's oxidized. NADH is oxidized into NAD+. Electrons are taken into complex 1, also called NADH dehydrogenase, because it's transferring hydrogens. And as it passes through, what happens is it powers up complex 1 to pump out protons. Okay? And then what happens here is ubiquinone, the energy carrier, goes to complex 1, picks up the electron. It's mobile, it moves. And then it travels to complex 3 and passes it to complex 3. So you see it's passing through, and as it passes through complex 3, it also energizes it to pump protons. And cytochrome C, energy carrier, that's what it does, it carries the energy. Electrons are energy. It also picks up the electrons and passes them to the next complex, complex 4, which energizes it as it passes through to pump more protons. And then as it travels down, it goes into the matrix. It's dropped off in the matrix to oxygen. Okay, so oxygen is the final electron acceptor, and it meets up with, actually creates a bond with hydrogen to form water. And now you have a lot of protons in the intermembrane space. And now, protons, they increase acidity. So right now it's really acidic in the intermembrane space. There's a high concentration. So this high concentration causes an electrochemical gradient. Two kinds of gradient. Electro because there's charge, and chemical gradient for the high concentration of protons. High concentration of protons. So because they are charged, and there are some electrons here in the matrix, they're attracted to the electrons in the matrix. So that makes them want to go down ATP synthase, diffuse down here through the membrane. Also, this high amount of protons also wants to go down its concentration gradient. Okay? So electrochemical, because there's charge, and chemical for the concentration gradient. Two kinds of gradients here. And so what happens as they go down ATP synthase, they power it up, make this bottom part here spin like a turbine, and as it's spinning, it takes the inorganic phosphate and adds it to ADP, turning it into ATP, and that's how you get ATP. Energy. The currency used for energy. Cell's currency for energy. Now, that's what happened with NADH. And now you have FADH, which doesn't go to complex 1. FADH2 goes to complex 2, and their electrons are given up to complex 2. So complex 2, as they go through it, also pumps out protons in the inner membrane space. And remember the energy carrier, ubiquinone? What it does is it moves over, it moves, remember it's mobile, it moves over to complex 2, picks up the electrons, and then sends them to complex 3, and they continue the same route, energizing complex 3 and complex 4 to pump out hydrogens, and then also to the same route, ending up in the matrix, and oxygen picks them up. That's why oxygen is the last energy acceptor, electron acceptor. Oxygen is the last electron acceptor. And then it forms a bond with hydrogen again, makes more water, and Again, we have the high concentration of the protons, which causes this gradient, causes them to go down, ATP synthase, causing it to spin, the bottom part to spin like a turbine, and that spinning causes it to combine the phosphate, inorganic phosphate, with ATP to produce ATP. Now, I have some practice questions too. Here. Number 1, the energy source that drives the protein complexes to pump protons into the inner mitochondrial membrane is the... Want to take a minute, pause the video? It's B, the flow of electrons down the electron transport chain. 2, the energy source that drives ATP synthase to synthesize ATP during oxidative phosphorylation is the D, electrochemical gradient that causes the protons to move across the membrane. 3, the final electron acceptor of the electron transport chain that functions in a aerobic oxidative phosphorylation is A, oxygen. 4, what is the main purpose of oxidative phosphorylation? A, to produce the most ATP. 5, electrons from which carrier are picked up by complex 1? C, NADH. 6, electrons from which carrier are picked up by complex 2? D, FADH. 7, when electrons flow along the electron transport chains of mitochondria, which of the following changes occur? The pH of the matrix. The pH of the matrix increases, A. So the matrix increases, that means the intermembrane space actually decreases. That's the matrix that's increasing. Remember here. The matrix here, the inside, that's where they're being pumped into. Going down into. So that is the matrix. Okay, and that increases. Their flow going down to the matrix makes the pH increase, but the intermembrane space will decrease. Remember the pH scale? Protons decrease the pH. Protons decrease the pH. And the opposite, hydroxyl groups increase it. So what happens at the end of the ETC, number 8? C, 4 electrons combine with a molecule of oxygen and 4 protons. Molecule of oxygen is O2. Okay, actually I will not be putting that question on the exam. Don't worry about it. That's how they make water anyway. What is the process for synthesizing ATP that uses the movement of ions across a selectively permeable membrane down their electrochemical gradient called chemoosmosis? And that's it for this video.